Greetings to everyone. Welcome back once again to this educational channel on biology. In this video, we'll be discussing the SBM Biology trial papers, that is Melaka Paper 1, Objective Paper for the year 2021, and this is of the new KSSM syllabus. So this video is the first part of two videos, and we will discuss questions 1 to 20. In part 2, we'll discuss question 21 to 40. So in this part 1, the questions are related to your Form 4 topics, okay, of the KSSM syllabus. And in part 2, questions 21 to 40 are related to the Form 5 topics. First question. Diagram 1 shows an animal whose scientific name is Crocodylus niloticus. So name the direction A as shown in diagram 1. Okay, so it's pointing to the underside of the animal, the belly partner. All right, and what is the view shown here? Yes, it is the ventral view. Okay, so what are the other views that we have? Now if you look from the top, from the upper side or the back of the animal, is called the dorsal view, just the opposite of the ventral view. And then if you look from the front, what is the view called? Anterior view. Okay, and you look from the back, the end, the end of the tail end is called the posterior view. Okay, right, let's go on to the second question. Question 2. Which of the following structures can be observed in both animal and plant cells using a light microscope? So there are two conditions here. The structure must be able to be seen in both animal and plant cells, and that is by using a light microscope. So it must be a big structure, and also we don't need to use an electron microscope to look at this structure, to see this structure. Okay, so let's identify the four organelles here first. Okay, A is a spherical or round shaped structure. Here is a round shaped structure, and it has a black dot in the center. So this is the nucleus, and the black dot is the nucleolus. Okay, now for B, what is structure B? Yes, structure B is rough endoplasmic reticulum. You can see the series of tunnels, and then it has the black dots on it, which are the ribosomes. Okay, so C, what is C? Now C has a double membrane, right? And then you can see all these structures inside. So what is it? Now the structures are called the grana, and then this may be all the food reserves, okay, the white structures here that are stored inside this organelle. So since it has a double membrane, right, which are the inner membrane is not folded, there's the outer membrane, inner membrane, so it's not folded. This structure is the, yes, chloroplast. And the fourth structure, you can see that it's also got outer membrane and inner membrane, and the inner membrane is folded to form the cristae. These are the cristae. So this is the mitochondria, correct? Okay, so now you've identified the four organelles. You must eliminate them, okay, if they don't fit the requirement. Now, first of all, which one cannot be found in both animal and plant cells? Yes, it's the chloroplast. Chloroplast is only found in plant cells, not in animal cells, because it's involved in the process of photosynthesis. Okay, so it's found in the plant cells, but not in animal cells. So C is out. C is out. And then, which can be observed using a light microscope. So you have seen the chick cell under the microscope, and one organelle that's very obvious is the nucleus, which is like a dense grayish shape, right? Not a round shape. Okay, so nucleus is the one that can be seen under light microscope, and we cannot see the rough endoplasmic reticulum, neither can we see the mitochondria, which are quite tiny, and also these structures can be seen only under the electron microscope. Okay, so the answer is A. Question 3. In an experiment conducted on the cell sac of a seaweed's vacuole, it was found that the calcium ion concentration in the vacuole was 330 milligrams per decimeter cube while the calcium ion concentration in seawater was only 13 milligrams per decimeter cube. What is the process contributing to the changes in the calcium ion content of the seaweed's vacuole? Okay, what is the process that contributes or causes the changes in the calcium ion content? Okay, so if we make a rough sketch here, if we sketch it, is like this. Inside the cell, you have the calcium ion concentration equal to 330 units, and then outside is 13 units, right? So now you can see that the cell has accumulated the calcium ions against the concentration gradient. That means it's taking in the ions, all right, from region of low concentration to region of high concentration, like climbing uphill. That needs energy, all right? And that means that the process is active transport, okay? So explanation, unequal concentrations of calcium ions inside and outside the cell implies that the cell is taking up or accumulating calcium ions against the concentration gradient, all right? So it needs energy to do that, okay, like climbing up a hill, and thus active transport of calcium ions by the seaweed is involved. So whenever you see this condition, this situation where the cell has much more of the ion, okay, a higher concentration of the ion compared to outside, then you know that this is a process of accumulating the ion, okay? It can also be vice versa, where the cell has much less lower concentration of an ion compared to the outside. But in this case, it is accumulating the ion, okay, by taking it in against the concentration gradient. So it cannot be osmosis, because osmosis for water, the movement of water, and simple diffusion is not the right answer too, because in simple diffusion, equilibrium, dynamic equilibrium may be reached where the concentration of the ions inside and outside the cell should be the same or almost the same, okay? But in this case, there's a vast difference in the concentration of ion inside and outside the cell. Thus, it is active transport. Question 4. The graph in diagram 2 shows changes in the mass of potato strips when they are immersed in different concentrations of sucrose solutions, right? So this graph shows changes of mass of potato strips, okay, against the concentration of the sucrose solutions in grams per 100 ml. Okay, so for this type of graph, one of the main things we should do is to determine where the isotonic solution is, right? Now, the isotonic solution has a concentration that's equal to that of the cell set of the potato cells. And when the potato strip is placed in such a solution, there should be no change in mass of the potato strips, right? Because the rate at which water enters the cell and diffuses into the cell and out of the cell is at equal rates. So therefore, the value of the isotonic solution is here, where there is zero change in mass of potato strips, where the curve cuts the horizontal axis here. What is the value here? Roughly 2.5 or 2.6 grams per 100 ml sucrose solution. Okay. So based on the graph, which of the following concentrations of sucrose solution should be used so that a flaccid potato strip regains its turgidity, right? So a flaccid potato strip is one that has lost some water through osmosis, so it's soft. So what kind of solution are you going to place it in so that it will regain, it will become firm again, all right? Regains its firmness. Usually the word turgidity is used for cells, for cells to regain their turgidity, okay? And then the potato strip will become firm and hard. So for this to occur, we must place the flaccid potato strip in what type of solution? Is it a hypotonic, isotonic, or hypertonic solution? Yes, we must place it in a hypotonic solution so that water can diffuse into the cells from the hypotonic solution into the cells by osmosis. And then the potato strip will regain water and it will regain its firmness. Okay, let's read the explanation here. The concentration of the sucrose solution, which is isotonic to the potato cell sap, is about 2.5 grams per 100 ml. And it's the intersection between horizontal axis and the curve. For a flaccid potato strip to regain turgidity, we need a hypotonic solution, which is less concentrated than 2.5 grams per 100 ml. or 2.6 grams, okay, around there, per 100 ml, so that water can diffuse into the cells by osmosis, right? So out of these 4 choices, we can see that only A is the hypotonic solution, okay, because the value of it is less than 2.5 or 2.6 grams per 100 ml, okay? So out of the 4, A is the hypotonic solution, all right? B is the isotonic solution, C and D are hypertonic solutions, as can be seen from this graph. So A is the answer, and from the graph can see A is a hypotonic solution, falls somewhere here, right? As long as it's less than, the value is less than 2.5 or 2.6 grams per 100 ml, sucrose solution. And then we have the isotonic solution at the intersection, and after the intersection here, we have the hypertonic solutions, okay? So it pays to remember how it looks like on this graph, okay, this picture. Okay, so your answer is A. Question 5. Diagram 3 shows the formation of maltose from molecules Q and R. Okay, so this process is called a condensation reaction, where 2 smaller molecules combine to form a more complex molecule and also to form 1 water molecule, which is not shown here, right? Now, what are Q and R? So Q and R are actually monosaccharides that combine to form the disaccharide maltose, right? So let's recall back. Now we know that in hydrolysis, maltose breaks down to form glucose and glucose, right? So I've taught you the acronym MAGGI, M-A-G-G-I. Maltose is hydrolyzed to form glucose and glucose. But this is the reverse reaction. It's condensation. So we have to draw an arrow going backward like this, all right? Or we write it as a forward reaction, that means glucose plus glucose forms maltose. Glucose plus glucose, arrow back this way, forms maltose. That's condensation. So we can just reverse it, and we find that the 2 molecules of glucose are needed to form 1 molecule of maltose. Okay, so the answer should be A, right? So think of the acronym MAGGI for hydrolysis and just reverse it backward if you are tested on condensation. Question 6. Diagram 4 shows the lock and key hypothesis of enzyme action. Based on the diagram, what characteristic does this enzyme show? So let's look at this diagram of the lock and key hypothesis. This is an enzyme. It has an active site with a specific shape, and the shape of this active site must match the shape of the substrate. So the shape of the active site is complementary to the shape of the substrate, right? So that the substrates can bind to the active site, and then a reaction can occur between the enzyme and the substrate, right? Similarly, for this enzyme too, the shapes of the substrate matches that of the active site, right? So what's the characteristic that is shown by this diagram? It is that the enzyme is specific in its reaction, highly specific, meaning that each type of enzyme, like this one here, can catalyze only one type of reaction that involves only one type of substrate. The substrate with the round shapes, okay? It cannot catalyze reactions involving these 2 other substrates because the shapes of the substrates do not match or are not complementary to the shape of the active site. So that means that the characteristic that's shown by this diagram, okay, that is involving the lock and key hypothesis, okay, the characteristic shown is B. Enzyme reaction is specific, meaning each type of enzyme can only catalyze one type of reaction involving one type of substrate. Question 7. Diagram 5 shows the longitudinal section of a plant root. Which part of the root system carries out mitosis? Okay, so we have to identify parts R, Q, and P. Now R is at the tip of the root. it is not a part of the root, but actually it's the root cap. Okay, R is the root cap to protect the tip of the root, right? So in Q we have two parts actually. The lower part, okay, the part is labeled, that is colored yellow here, is actually the apical meristem, right at the root tip, where, and it's also called, this area is also called the zone of cell division, because the cells are dividing actively by mitosis, right, to produce new cells for the growth of the root. Okay, so your answer should be Q lah, right? But let's look at the other parts. So in Q also we have another part somewhere here, which is not separated out, okay, and that is zone of cell elongation. Okay, so after the new cells are formed, they have to grow, they have to enlarge and elongate. So this second zone, after the zone of cell division, we have zone of cell elongation. And lastly, when you see the root hairs forming, it means that the cells have started to differentiate and form new structures, okay, in order to carry out their functions more efficiently. Okay, so this is the zone of cell differentiation, P. So the answer is Q, where the apical meristem is found that carries out mitosis. Question 8. Fermentation of glucose by yeast can be shown by the equation below. What are substances P and Q, right? So we have to know the equation for fermentation of glucose by yeast, okay, where no oxygen is used to break down the glucose to form P and Q and energy. Okay, so what is the acronym that we can use for this equation? Yes, it is GC. So here GC stands for G, glucose, is broken down to form what? C for carbon dioxide, E for ethanol, and the other E is for energy, right? So your answer is, for P and Q, it will be C, ethanol and carbon dioxide. Okay, the C is for carbon dioxide, the E is for ethanol, right? Very simple if you remember the acronym GC for fermentation of glucose by yeast. Question 9. Diagram 6 shows a part of the human respiratory system. Which of the following are the adaptations of the structure for gaseous exchange, right? So first you have to identify this structure found in the human respiratory system. These are the alveoli, okay? And surrounding the alveoli are many blood capillaries. So what are the adaptations? Now let's go through the list here. It has a small surface area. No, okay? You should have a large surface area because there are many alveoli. And has a thin wall. Yes, the wall of the of each alveolus here is thin and is only one cell thick for faster diffusion of respiratory gases. So number 2 is correct. Number 3 has a moist surface. Yes, the internal surface of each alveolus is moist, right? So that the respiratory gases can dissolve in it. And then 4, covered with many blood capillaries. All right, so it has a network of blood capillaries, okay, on its outer surface here. So number 4 is also correct. So 2, 3, and 4, the answer is D. Question 10. Which of the following occurs during exhalation process in a fish? Right? So underline the word exhalation, not inhalation. This is for exhalation process. Now what happens to the mouth operculum and the flow of water in the exhalation process? So we must understand that in exhalation, the water is flowing out of the fish body. Whereas in inhalation, now you can see in inhalation, the water will flow into the mouth. So this is when the mouth opens. But in exhalation, right, the mouth has to close so that the water can be pushed out through the operculum, right? So mouth must close and operculum must open. so that the water can flow out of the operculum here, from under the operculum. Okay, so this red arrow is for inhalation, but for exhalation the mouth must close, then water will flow out from out of the through or under the operculum. Okay, so answer is C. Question 11. Diagram 7 shows a part of the human digestive system where the digestion of food starts. Which of the following statement correctly describes what happens in the mouth? So let's go through the answers. Saliva emulsifies fats into smaller droplets. So saliva doesn't emulsify fats, okay? It doesn't have this action to emulsify fats. So actually what emulsifies fats is the bile from the gallbladder that's secreted into the duodenum. So this is wrong. Chewing increases the surface area of food for digestion. Correct. So in the mouth, we chew our food to increase the surface area of the food, because the food will be broken down like a piece of bread. When it is chewed, it is physically broken down into smaller pieces. All right. Then the surface area of the food will increase so that the enzyme can act on it. The enzyme amylase can act on it at a faster rate or higher rate. So number 2 is in. Teeth break up smaller soluble molecules into large insoluble molecules. Teeth break up smaller soluble molecules into large, from small to large. So that's not breaking down, right? And actually, the molecules are not broken down by the teeth. The teeth just physically break up the solid particles, a solid piece of food into smaller pieces. So this one, smaller to large, is already wrong. Okay. So next, salivary amylase breaks down large starch molecules into smaller maltose molecules. Okay. So salivary amylase is an enzyme. It breaks down the large starch molecules chemically, okay, through hydrolysis, right, to form smaller maltose molecules. This is correct. This is the process of digestion or hydrolysis carried out by the enzyme salivary amylase. So answer is 2 and 4. So answer is C. Now question 12. Diagram 8 shows a longitudinal section of the human heart. Structure R is a pacemaker. What is the function of R? Right? So what is the exact scientific name? The scientific term for R? Okay, the name is sinoatrial node, or SA node. So what is the function of R, or the function of the sinoatrial node? Right, we know that it helps to generate impulses, right, from it. And these impulses will flow to the walls of the atria, the left and right atria here, and the left and right atria will then contract together. So that's the function of the sinoatrial node. So the answer is B. So here I write the sinoatrial node generates electrical impulses which spread rapidly in both atria, causing both atria to contract and pump blood into the ventricles. Okay. Question 13. Diagram 9 shows structure X in the human lymphatic system. Which of the following is the function of structure X? Okay. So we have to identify what X is. So it's all these round little nodules found in the lymphatic system. So what is X? Yes, X are the lymph nodes. Okay, X are lymph nodes. So what's the function of the lymph node? Now we know that in the lymph node there are phagocytes and lymphocytes, which are white blood cells that help to destroy bacteria and foreign bodies. And so in that way, they filter the bacteria from the lymph that flows through them. Okay, so the answer should be, yes, C, to filter bacteria and foreign bodies from the lymph, so that the lymph has less of these pathogens, okay, and foreign bodies in it for defense, as a part of the defense mechanism. Question 14. Diagram 10 shows the concentration of antibodies in the blood after 2 injections. Right, so let's read the graph. Concentrations of antibody in the blood against time. The shape is like 2 hills, okay, for the graph. So this tells us that the substance injected into the person is a vaccine. And vaccine, what is it? Vaccine contains a suspension of dead or weakened pathogens that are injected to stimulate the lymphocytes in a person's body to produce antibodies against a specific disease, right? Now, which of the following statements is true about the graph? The individual is injected with antiserum from another individual for the first and second injection. So what's an antiserum? An antiserum contains the antibodies that are directly injected into a person, all right, especially a person who may have already been infected. So here is not antiserums that are injected, okay? They are vaccines. Vaccines are injected into the person. So this statement is wrong. I should be the individual is injected with a vaccine. Now for B, both injections contain living pathogens which control the production of antibodies. So the word control here is wrong. Now both injections may contain living pathogens because they are vaccines, okay? Living but weakened pathogens, right? But these pathogens don't control the production of antibodies. They stimulate the production of antibodies by the lymphocytes, okay? C, second injection is required to boost immunity. Cancel this word level. Second injection is required to boost the immunity of a person. Correct. So you can see here that after first injection, the level of antibodies is still very low. But after second injection, the level of antibodies rose very quickly, okay, at a faster rate and in greater amounts, all right? It rose very quickly above the level of immunity, right? So this will make a person immune at this stage here, around nearly 6 months. When the curve cuts the line for the level of immunity, all right? So second injection is required to boost the immunity of a person and to make the person immune, okay, in this case. Now, so the answer is C. Let's look at D. Second injection contains a higher level of antibodies. No, the injection does not contain any antibodies, okay? In the injection, the substance that's injected into the body is a vaccine, which are dead or weakened pathogens, okay, not antibodies. So this choice is wrong. Question 15. The information below describes the functions of a part of the human brain. Controls voluntary movement like walking and jumping. Involves intelligence and learning, and for sensing the environment, okay? So the description refers to the structure A, B, C, or D. So identify all these structures first if possible, and then find out which one has these functions, right? So A is the cerebrum, and the cerebrum is responsible for higher mental functions like learning, right, and reasoning. So here we already have the function intelligence and learning. So the answer is definitely A, right? Whereas C is the cerebellum. It maintains body balance and muscle coordination. And D, now below D is called the pons, P-O-N-S. And below D here is the medulla oblongata, which is involved in carrying out involuntary actions, certain involuntary actions like coughing, controlling the heartbeat rate, and so forth, okay? B, we don't really study or label this part, okay, in our syllabus here. Okay, so the answer is A. Next question, question 16. Which hormone is correctly matched to its function? Right, thyroxine. Does it stimulate the absorption of water in the kidneys? No. Thyroxine, the function of thyroxine is to increase the rate of metabolism. Okay, and the hormone that stimulates the absorption of water in the kidneys is ADH, antidiuretic hormone. Okay, progesterone stimulates the development of the uterine lining in females. Yes, this is correct. So progesterone will stimulate the endometrium, okay, in the uterus, which is a layer of blood and tissues, to develop, okay, in the menstrual cycle, during the menstrual cycle. So B is the answer, right? Now let's go on to the others. Testosterone stimulates conversion of glucose to glycogen. Definitely wrong, okay? So testosterone is responsible for stimulating spermatogenesis and development of the male secondary sexual characteristics, like the growth of mustache and so forth, okay? And the hormone that's involved to stimulate conversion of glucose, excess glucose to glycogen, is insulin, all right? And then lastly, estrogen stimulates ovulation in the ovary. This is not correct, because estrogen has a similar function to progesterone. It repairs and stimulates thickening of the uterine lining after menstruation, right? So the hormone that stimulates ovulation in the ovary is actually luteinizing hormone, or LH. So our answer is B. Question 17. Which of the following is part of the homeostatic process? So first you have to know what's the meaning of homeostasis, right? The definition is that it is the regulation of physical and chemical factors of the internal environment within normal ranges, okay? And the physical and chemical factors are, for example, temperature of the body, okay, blood osmotic pressure, right, and also concentration of glucose in the blood, and also the blood pH, right? So when these factors are being regulated, then we say that homeostasis is being carried out, right? So let's look at the answers. Contraction of circular muscles during peristalsis. This doesn't regulate any physical or chemical factor. Focusing of the eye on a distant object. Again, it doesn't regulate any physical or chemical factor, right? And then secretion of digestive enzyme. No, this is just the activity of the glands that leads to digestion, right? Okay, let's look at the vasoconstriction of arterioles in the skin, right? So this process helps to regulate the temperature of a person when a person is feeling cold. So when a person is feeling cold, the arterioles in the skin will constrict. It sort of contracts, so that it becomes smaller. The lumen becomes smaller. Okay, so we call that vasoconstriction of arterioles. Okay, so that less blood flows to the surface of the skin and less heat is lost to the environment through radiation. So it will cause the person to retain the heat in the body, and this is to conserve heat, so that the temperature of the person will not drop so much. So this is a type of homeostatic process to regulate the body temperature. Okay, so D is the answer. Question 18. Diagram 11 shows ultrafiltration that occurs in the kidney. Why are there no erythrocytes found in X? Okay, so let's look at this diagram. Now this structure inside is called the glomerulus. Inside the cup-shaped structure is called Bowman's capsule. All right, so the blood is flowing into the glomerulus via the afferent arteriole and out of the glomerulus via the efferent arteriole. All right, so we can see here that some fluid has been pushed out, okay, because of the high hydrostatic pressure in the glomerulus. Is pushed out into the Bowman's capsule, right? So why are there no erythrocytes in X? Now, I believe there's a mistake here. This should be, X should be here, all right, the Bowman's capsule. Why are there no erythrocytes in the Bowman's capsule or inside, in the glomerular filtrate inside the Bowman's capsule, okay? So the fluid that's formed here is the glomerular filtrate, right? Actually comes from the glomerulus, okay? So I'll rephrase the question as, why are there no erythrocytes found in X, which is the glomerular filtrate, okay, that's found in the Bowman's capsule? Now, so what is the answer? The answer is that the erythrocytes are too large to pass through the capillary walls. So they remain inside the glomerulus. They don't come out because they are too large to pass through the pores in the capillary walls. Okay, so there are no erythrocytes in the Bowman's capsule here. So what is the right answer? Now let's look at the answer. It is destroyed in the liver. It is a large molecule. It is used up by body cells for cellular respiration. It is less abundant in the afferent arteriole. So the closest answer is B. But erythrocyte is not a molecule. It is much bigger than a molecule. In fact, it's made up of many molecules, right? So erythrocyte is a cell. Therefore, we can say the answer should be erythrocytes are too large to pass through the capillary walls. They are too large to pass through the capillary walls. And so they remain inside the glomerulus, inside here. Question 19. Which of the following animals has a hydrostatic skeleton and an exoskeleton at different stages of its life cycle in a complete metamorphosis? So let's highlight the keywords: hydrostatic skeleton, exoskeleton at different stages of its life cycle, complete metamorphosis, right? So this word complete metamorphosis really gives us an idea which animal it is, all right? It's not the leech, neither is it the crab, because these don't undergo metamorphosis, right? So is it the grasshopper? Now grasshopper undergoes incomplete metamorphosis, not complete metamorphosis. So grasshopper is out. And the answer is the fly that carries out or performs a complete metamorphosis, where there is a larva and a pupa stage. Grasshopper, it doesn't have a larva or pupa stage. It has the nymph, the stage called the nymph, and there are many nymphs at different stages of its life cycle. Sometimes they call it the instar. Right, let's come back to the fly. Right, so let's have a look at the life cycle of a fly. Now, the adult housefly lays her eggs. After that, the eggs hatch, and out comes the larva. So the larva will go through a few stages. Now, it is at the stage of the larva, okay, where it has a hydrostatic skeleton, just like an earthworm or a leech. It's a soft type of skeleton. Okay, actually, a hydrostatic skeleton is a fluid-filled body cavity where the fluid is under pressure, and it is surrounded by a muscular body wall like this. All right, this is the fluid-filled body cavity in the center of the body, and then it's surrounded by a muscular wall which consists of, for example, in the earthworm, it consists of the circular muscles and the longitudinal muscles, right? Now, so this is the hydrostatic skeleton. So it has this hydrostatic skeleton when it is a larva. After that, the larva will move into the pupal stage, and there the transformation occurs where it changes into an adult housefly with wings. Okay, so when the pupa comes out, you have the adult housefly, and this adult housefly has an exoskeleton. So it has a rigid outer covering made up of chitin here, and this is its exoskeleton. The soft body tissues are inside. All right, so exoskeletons are also found in mollusks, like the snails, okay, that build their shells using the calcium salts. Okay, but the insects do have exoskeleton. Okay, so the answer is A. Right. Question 20. Which of the following figures shows how fraternal male twins are formed after fertilization? Right, highlight this word, fraternal male twins. This is our keyword, right? These are our keywords. So fraternal, first let's discuss what's the meaning of the word fraternal. A pair of fraternal twins are twins formed when two sperms separately fertilize two ova, right? So look for the answer that has two ova. Okay, the circle with the dot here is an ovum. So only C and D fits the bill. They have two ova each. Okay. Next, male twins. So male twins are formed when there is one X and one Y chromosome in each of their cells. For males, they have one X and one Y sex chromosome. Okay, right. So the ovum, firstly, you must know that each ovum only carries one sex chromosome, one type of sex chromosome, which is the X sex chromosome. Okay, all ova carry one X sex chromosome each. There's none that carry Y chromosome, the Y chromosome. Okay, only the X chromosome. So that means to get a male twin, the sperm must carry the Y chromosome. Okay, because there are two types of sperms, one that carries the X chromosome and one that carries the Y chromosome. Okay, so in this case, if the two twins, two of them are males, that means both the sperms must carry the Y chromosome. So that when it fuses with the ovum that carries the X chromosome, all right, in this case, then a male with the pair of chromosomes XY in each of his cells will be formed. Alright, so if you look at C and D, only D fits the bill. Each sperm carries a Y chromosome. So both these zygotes formed after this will be male. Whereas for C, you have one sperm which carries the X chromosome here. So if it fuses with the ovum, we'll get the pair of sex chromosomes XX in each of the cells, and that will form a female or a girl, not a male, right? So the answer is D. Right, that's all for this lesson. Thanks for watching. Please share, like, and subscribe, and goodbye for now.